Welcome everyone, and thank you for joining us for our presentation titled Eliminate Temperature Sensitivity and Lyophilization Technology. It's my pleasure to introduce you to Amy Webster, a custom manufacturing service leader with genomics and diagnostic solutions at Cytiva. For a complete biography of our speaker, please visit the tab at the top of your screen. Now, if any questions arise during the presentation, we encourage you to submit them in the Q&A box. Our speaker will address your questions following the end of our presentation. Please join me now in welcoming our presenter, Amy Webster. I will now turn the presentation over to her. Welcome, Amy.
So thank you very much for the kind introduction. We're here today to talk about how to eliminate temperature sensitivity with lyophilization technology. So first off, I'm going to give you a brief introduction as to who I am and who Cytiva is, and then we go through the principles of lyophilization, how you would do it, what you can use it for, and why you would do it. And I can then go through a case study for you and give you some further information and links to videos of the site, and then go through any questions. So introduction. My name's Amy, and I work at the Cardiff manufacturing site for Cytiva in the UK. I am part of a team called the Customized Solutions team. We're a team of 7 dedicated scientists, and we are entirely dedicated to customer projects, and we have no R&D responsibilities. We have 5 main services, what we call plates, conjugation, contract manufacturing, custom biology, and lyophilization. So the contract manufacturing, we can take your process and we can manufacture it in-house. Plates, we've got a range of filter plates that we can put custom membranes in. Custom biology, we can take any of our catalog products and customize it to your requirements. Conjugation, we have a range of magnetic beads that we can conjugate antibodies or oligos to. And Lyo-Stable is our freeze drying and lyophilization service, which we're going to go through in more detail. So lyophilization, how do you do it? What can you do it to? And why would you do it? So lyophilization is a stabilizing technique which takes a liquid, freezes it, sublimes all the water from it, and leaves you with a solid glassy cake. With the cake being a solid, the reagents in it can no longer move, and there should be no side reactions and no degradation occurring in the solid state. Benefits of lyophilization. So why would you want to lyophilize? One of the big advantages of lyophilization is ambient storage and shipment. This means that you no longer have to ship enzymes and other reagents around the world on dry ice and ambient conditions. Everything can go in the same package. It can be stored at point of use at ambient. You don't need a -80 and -20 and a fridge at the same time. And this saves not only in dry ice and benefiting the environment, but also as the shipper, it saves an awful lot of money being able to ship at ambient rather than needing dry ice. It also significantly simplifies the workflow at point of use. All of the formulation has been done in one big batch beforehand and has been QC tested. So each one of the lyophilized pellets or cakes has the same activity and has already been tested. The end user only has to add the sample and then run the experiment. This means that the end user can run multiple different tests using exactly the same protocol by just changing the lyophilized cake. With a big batch size, you only have to run one QC for the entire batch, and your repeatability and reliability of your assay will be improved. And the simplification of the workflow at the point of use also means that the waste is reduced of not only the raw materials, but also of the plasticware and tips. There is less pipetting steps, there is less freeze thawing of the enzymes. The mix you use on day 1 will be the same as the mix you use on day 5. So what type of assays can we lyophilize? Most types, and if we've not done it, we'll definitely give it a go. So we've done LAMP assays, qPCR, RT-PCR. With that, we've done many enzymes. We've done RNA for QC analysis and NGS sequencing. We've got a range of catalog beads, which do genomify and templify, which are rolling circle amplifications. We've lyophilized conjugated mag beads, and we've also started lyophilizing a range of exosomes and biological reagents along those lines. So how do you design your assay for lyophilization? 3 main points, and we're going to go through each of them. The first one is excipients, second one is formulation, the third one is what format you want to put it in, cakes or beads. So first off, the excipients. Now, excipients are very important for lyophilization for 2 main reasons. One, they provide the structure for the cake, which aids in the reconstitution and also the stability. But more importantly is they protect any delicate materials that are in the cake. Enzymes and proteins have a complicated folding structure. And in a solution, this folding structure is held in place by the hydrogen bonds from the water molecules. During lyophilization, you'll be removing the water molecules. So you need to replace those intramolecular bonding with something else, otherwise the protein is going to denature. And it's the excipients that take over this. It's the excipients that take over this bonding and retain the activity of the enzymes. Cytiva has developed a range of different proprietary excipients that are compatible with many different applications and have proven to give stable product. One thing with excipients is excipients can interfere with your downstream assay. So the less excipients that we have to put into the lyophilized cake, the better. The main way we do this is if you had a, say, 50 microliter PCR, we would dispense the reagents and excipients in 10 microliters. We would lyophilize a 10 microliter cake or a bead, and then at point of use, it would be reconstituted in 50 microliters, which would significantly decrease the amount of excipient in the downstream applications. The next important thing is the formulation. You're about to spend hours removing all the water from the solution. So if you can concentrate the raw materials and reduce the amount of water you add, you have a lot less water to remove. Things like changing primer concentrations from 10 micromolar to 1 millimolar, magnesium concentrations to 1 molar, and things like that. There is a limit to the amount you can concentrate your reagents, and normally this normally is limited by the salt concentration. Now, as you know, they spread salt on the rows to prevent it freezing, so increasing the salt concentration in your assay will decrease the freezing temperature. The more you decrease the freezing temperature, the harder it's going to be to lyophilize. There are a few other things that lyophilization can't cope with, the main one being glycerol. So for lyophilization, we often have to source either no glycerol or very high concentration enzymes so that we really limit the amount of glycerol going into the solution. Another solvent that we don't like is DMSO. And again, too much DMSO will prevent the lyophilization from being successful. The plasticware that is used to lyophilize is also very important when you think about the cycle. The design, whether or not the plasticware is going in the freeze dryer, and the shape of the plasticware will significantly impact the ability to lyophilize. We also need to be able to properly seal the plasticware so that after we've dried the cake, we can maintain that in a proper environment. And we'll go into slightly more detail about that later on. The next thing that we need to decide is what format we want it in. Now at Cytiva, we have 2 formats which we call cakes and beads. Both of them use the same proprietary excipient mix, and both of them use the same processes. The difference being the order at which they are lyophilized and put into plasticware. So for cakes, we'll do the formulation. We will then dispense into the final plasticware, and then we will lyophilize in situ in that plasticware. With this method, we put in the plasticware into the freezer, so the plastic needs to be able to cope with the freeze drying cycle. And because of this, our batch size is limited to about 20,000 reactions. For the bead format, what we do is we do the formulation, we then drop and they form spherical beads, and we lyophilize in those spheres. We then pick and place those beads into the final plasticware and seal those and pack those. Our typical batch size for beads is anywhere from 5,000 to 350,000 reactions. So both formats can use pretty much any plasticware, tubes and vials that you'd like, that can be sealed. So going through stages of a freeze drying cycle. So the first thing we need to do after we put the after we've done the formulation and dispensed it is to freeze the mix. This is important because the next step we're going to do is put a vacuum, and if the mix is not properly frozen, then the vacuum will pull the liquid out of the tube and we will lose the entire cake. One thing we can do in the freezing step is to use annealing steps which cycle between solid and liquid. This will form bigger ice crystals which would make drying easier. The next stage is primary dry. In this, the vacuum is pulled, so it will pull the conditions in the chamber below the triple point. This causes ice to sublime from a solid into a gas and dries the cake. During this stage, all of the unbound water is removed from the cake. The length of the primary dry is very dependent on the height of the cake. So the cake dries from the top down. Water is removed from the top layer, and then as you can see in the second and third pictures, the cake proceeds downwards the tube. So tall, thin cakes will take a lot longer to dry. The temperature of which the primary dry is set at will depend on the salt concentration and the formulation. In general, higher salt concentrations need lower temperatures to dry. The lower the primary dry and the greater the vacuum that's pulled, the longer it will take to dry. After all of the unbound water has been removed in the primary dry, we then move on to the secondary dry. A lot of freeze dryers can have endpoint determination tests at the end of the primary dry, which will determine when this is. So they can be used to automatically extend cycles where needed. In the secondary dry, the temperature in the chamber is raised and the bound water is then removed. Again, at the end of this stage, a lot of freeze dryers can do endpoint determinations and confirm that no more water is coming off. After the cycle is finished, the freeze dryer then goes into a storage phase. This effectively stops anything else from happening and allows the cycles to be repeatable day after day. In the storage phase, the machine's basically just holding everything in status until it is ready to be unloaded. So you've just spent a day or two removing all of the moisture from your cake. We then need to pack it in a way that it's going to maintain its dryness. Any extra water, any water that the cake absorbs is going to reduce its stability. So the stability of the cake comes from it being a solid. None of the reagents can move and start interacting with each other, but any water ingress into the cake is going to increase the mobility of the reagents in there, allowing degradation or side reactions to start happening. So the primary seal that we put onto the cake, it can either be done in the freeze dryer by means of collapsing shelves and driving bungs into vials, or we can move them into either a nitrogen box or a dry box, which are held at very low humidities, so under 1% humidity, and then we put a foil seal on. The cakes that are then sealed, we then pouch into a desiccated foil pouch, with whatever label is the customer requires on there. And this step is done in a room that's held at under 35% humidity. So if stored in the sealed foil pouch, then our typical stability is for well over a year for our custom products. So going on to a case study. So earlier on this year, there was a company called GeneDrive, who are a point-of-care diagnostic company. They came to us and asked us to help to work with them to develop a COVID test that could be used in labs around the world. So just going to talk through, in 3 months, we managed to design, develop, and then transfer a COVID assay. So for the first stage, we sat down and planned out the project with GeneDrive with their requirements that the tests would be able to be used in labs worldwide. This meant that it would have to be able to be used in multiple different thermocyclers. Now, a lot of different thermocyclers need different tube types. And to prevent having to transfer from, after they've reconstituted the cake or the bead, we did not want to have to transfer that reconstituted reagent into another tube to be able to run. So this meant that we would have to have many different plasticwares for this product. As it was a COVID test, the forecast was also very big and the demand would have been high. Because of this, we decided on the bead format. Now, the first steps we did were some initial proof of principle runs in the dedicated R&D facility. So at Cardiff, we have 2 facilities, one's dedicated to R&D and one's dedicated to manufacture, both of which have the same, or pretty much the same equipment, but the R&D version is slightly smaller. So with GeneDrive designed the test and sent us down the reagents. We lyophilized several different versions of it, sometimes in the same run, sometimes in different runs, took them out the freeze dryer and sent them back up to GeneDrive as quickly as possible. We then sit down and go through the results with GeneDrive, decide on the next steps. So after several iterations, we came upon a formulation that was fixed, we were happy with, and we then looked to transfer this into manufacture. So during the transfer process, what we wanted to do was ensure that we had a robust product and that we'd be able to manufacture it day in, day out. So we worked with GeneDrive and decided that we would run 3 different batches and we'd slowly scale up from an R&D size run to a manufacture size run. Now, the idea of the product with GeneDrive is that we would manufacture 1 batch of bulk beads. And these bulk beads could be stored at Cytiva and then dispensed into the plasticware needed. So there are 4 different types of plasticware, depending on which thermocyclers would need to be used. And we'd be able to use the one batch of bulk beads to dispense into any of these plate types. After we'd run the transfer runs, we sat down again with GeneDrive and confirmed that we were happy with the results, they were happy with the results, and that with the transfer had been successful, and this product was now launched in manufacture. So currently it's in manufacture. The manufacturing team run this regularly, and because of the way the assay has been designed, when GeneDrive have discovered that a further plate type is needed, then it's a simple case of validating the new sealing methods for the plates, but the bulk beads remain the same, so we can launch new products quickly. So to summarize, GeneDrive came to us at the beginning of the year wanting to develop a COVID test. We worked with them to streamline the test, push it through our systems as quickly as possible while covering everything that we needed to cover. And we now have a manufacturing product which is suitable for COVID tests around the world in many different thermocyclers. So we've got a couple of places where we can get more information. There is a video which shows you our Lyo-Stable facility and takes you on a tour around our lab. We've got a couple of papers that are useful, and we've also got a form we also have a form for you to request a proof of principle run. Thank you very much. And is there any questions?
Thank you, Amy, for that outstanding presentation. We will now move to the live Q&A portion of the presentation. As a reminder to the audience, please submit your questions via the Q&A box. So let's take a look. It looks like we already have some great questions coming in, Amy. Our first question, how long does it take for a proof of principle to be run?
So the first thing we'll do is work with you to decide on exactly what we want to test, both what fits you best and what we can run in one run. And after we've decided that and fully locked down, the aim is to have samples shipping back out to you within 2 weeks of receiving your reagents on site for a standard run. If it's a larger run, and particularly if it's a bead run, this may be a bit longer, but we'll let you know beforehand if it will be any longer.
Wonderful, thank you. And now, how many excipients do you have?
So we do have a range of excipients. We have 8 standard ones if we need them. Typically, we would start off with up to 4, depending on the type of assay we're lyophilizing. Initially, we test them wet, and we try 1 or 2 in the first few proof of principle runs.
Thank you so much. One audience member says, I am ISO 13485 regulated. Can you manufacture our products?
Yes, we can. So we are currently ISO 9001 regulated in Cardiff. We are in the process of getting 13485, but we don't have it yet. And we do manufacture many 13485 products.
Thank you so much. Now, how much does a proof of principle run cost?
So the ethos of the custom team is not to make any money off the proof of principle runs. We look to cover our costs. So our proof of principle runs start from as little as 1,500, and they go up depending on what the complexity of the POP and the number of samples needed.
Thank you, and I want to thank our audience members for these great questions. Our next question is, do we have to do any validation on ourselves as part of the proof of principle?
So for the proof of principles, there would be no validation that we would need to do. However, after the aim of the proof of principles is to get a product into manufacture. And as part of the manufacturing process, what we need to do is scale up. So we need to start looking at things like how we dispense. So in the proof of principle runs, we'll be dispensing 30 plates at a maximum probably.
Thank you so much. Is it possible to arrange a site visit to the Cardiff site?
Yeah, definitely. Normally, we say yes. At the moment, we're doing a lot more virtual tours, so we can give you a virtual tour of our site and we can do virtual audits, and we can still welcome visitors if we need to. Normally, yes, we're very open to visits.
Wonderful, thank you. We have time for one more question. What is the lead time, Amy, to secure a GMP run?
So it does depend on the complexity of the project. So once we've done all the proof of principles and we have a lockdown formulation, we will then start the transfer runs. Now, this can take anything from 6 weeks to a couple of months or longer, depending on the complexity of your project and the urgency of which everything is needed. We'll take it through to the core team. So once we've done the development, we then report our findings to the head of R&D, head of manufacture, head of tech support. And between us and working with yourselves, the customer, we will develop a action plan as to checking out all the risks that we need to look at before we can put this into manufacture. Once we've done the design transfer plan and we've validated everything that we've agreed on, we will then need to set it up in our systems so it So it is manufactured as a full-on product. So it can be anything from a couple of months to longer, depending on complexity.
And Amy, this same audience member wants to know, is it possible to get a copy of this slide wheel of services provided?
We have a copy of the information. So in our booth, the Accelerating Diagnostic Development with Custom Design Services, there is a collection of web links. One of those for the custom services for genomics is links to a web page which has all the information that was in that slide.
Wonderful. Amy, thank you for your presentation today and for your important research. Would you like to offer any closing remarks to our audience before we go?
Just thank you for listening. And if you have any further questions, please join me in the booth.
Amy, thank you again, Amy Webster, and I want to thank our audience members for their participation. Any questions that were submitted and not answered today by our speaker will be addressed via email. This presentation will be available for on-demand viewing for up to 12 months, so please remember to share it with your colleagues who might be interested in today's topic. And don't miss out on the other presentations on our agenda. Visit the agenda tab at the auditorium for a full listing. Thank you again for your participation. Until next time, take care, stay healthy, and stay safe. Bye-bye.